This lesson is on chromosome structure and will serve as an introduction to mitosis, which we'll cover in an upcoming lesson. Here are your objectives. So first, mitosis is the division of the nucleus into two genetically identical daughter nuclei. It's not the division of the whole cell, and that's often a mistake that people make, but it's the division of a nucleus into two genetically identical daughter nuclei. Now, meiosis, which we will also learn about in an upcoming lesson, is the division of the nucleus into four genetically distinct daughter nuclei. So there's a difference between the two right there. Mitosis occurs in eukaryotes only. Remember, mitosis is the division of the nucleus, and only eukaryotes have nuclei, so no mitosis in prokaryotes. And mitosis is involved in the production of all cells except gametes. Gametes are sex cells, sperm and egg cells, and they're produced by meiosis. So mitosis produces everything else. Mitosis is involved in growth, how an organism grows from a fertilized egg, a zygote, into a complex multicellular organism, and in repair lifelong. The separation of the cytoplasm is not, strictly speaking, part of mitosis. It's separate, and it's called cytokinesis. Now, to understand mitosis, we need to first make sure that we have our terms about chromosome structure clear. Let's talk about that. So here is a drawing of a chromosome. And what I'm showing you here is one DNA molecule. It's one DNA double helix. And it's in its condensed form, which is going to happen at the start of mitosis. But it's easier to think of it this way, to imagine that it's one single molecule. It's often more unspooled, kind of like that, all over the place. But we're going to think of it this way, just to help us understand what's going on. Now, DNA replication, which does not happen during mitosis, but happens separately, again, we'll talk about that when we talk about the cell cycle later, leads to a chromosome that has two DNA molecules in it. So this now is two DNA molecules, two DNA double helices, again, very condensed. And what you're seeing here are two sister chromatids. They are genetic copies of each other, and they are joined. This is one here, and this is one here. They are joined at the center here at the centromere. And again, the two sister chromatids have been formed in DNA replication from what was a one chromatid chromosome. So we've gone from a one chromatid chromosome here, a one chromatid chromosome, to a two chromatid chromosome through DNA replication. And we're going to use those terms, one chromatid chromosome and two chromatid chromosome, to help us keep things straight. Okay, so DNA replication takes us from a one chromatid chromosome to a two chromatid chromosome. Again, when DNA replication is happening, the chromosomes are actually not condensed like this, but they show up condensed like this the first time we see them in prophase, and we'll talk about that in mitosis. Now, just a few more terms. This part here, this could be called the long arm of the chromosome, the way I've drawn it, and this would be the short arm. Just visually, one is longer than the other. And again, these are sister chromatids. Now, in mitosis, this is going to separate out, and not drawn to scale, we're going to end up with the two sister chromatids separating out into different nuclei. So DNA synthesis has taken us from a one chromatid chromosome to a two chromatid chromosome where the sister chromatids are identical copies of each other, and then mitosis takes us back to a one chromatid chromosome in each nucleus, this being two different nuclei. Let's talk about one more important piece of terminology. So a cell with two copies of every chromosome, one maternal copy and one paternal copy, is called diploid, while a cell with only one copy of each chromosome is called haploid. So humans have 23 pairs of chromosomes, but they have 46 total chromosomes, because in each pair they have one maternal copy and one paternal copy. So 46 is the diploid number for chromosomes. That's written like this: 2n, n representing one complete set of chromosomes, 2n is equal to 46. And n is equal to 23 would represent the haploid state for a sperm or an egg cell. We'll talk about that in more detail when we get to meiosis. So again, a cell with 2 copies of every chromosome, 1 maternal and 1 paternal copy, is called diploid, and a cell with only 1 copy is called haploid. Now be careful, a common error is mistaking what's going on here, what we've shown above, as going from haploid to diploid, and that is not true. What's happening here, let's say we're looking at chromosome 6, is one parental copy of chromosome 6 duplicating itself, which later separates. There would be the other parental copy of chromosome 6, which we haven't shown, which would be doing exactly the same thing. And so what we've shown now is the complete pair, called the homologous pair, with one copy from each parent, each duplicating itself, and then each would later be separating out. And we'll show all of that stuff when we get to mitosis. But just be aware that here we have not changed the ploidy. We haven't gone from haploid to diploid or vice versa. Thinking we have is a common mistake people make. At the start of mitosis, the parent cell is diploid. And at the end of mitosis, the 2 daughter cells are still diploid. So there's no change in the ploidy being what we use to describe the state of being either haploid or diploid. There's no change in the ploidy in mitosis. Okay, so that's an introduction to chromosome structure, serving as the first step in thinking about mitosis.